Welcome to the third video of the Membranes and Cell Compartments unit. This video will describe the differences between prokaryotic and eukaryotic cells, the role and function of key organelles, and the mechanism of protein sorting required to get proteins to their correct cellular locations. All of the different organelle functions discussed in the video are able to occur because of the various proteins found in each location. For example, lysosomes can digest cellular debris because they contain digestive enzymes, while the mitochondria is able to produce energy because it contains ATP-producing enzymes. In order for these different organelles to work properly, translated proteins must get to their proper cellular location. The process of getting proteins to where they need to be to function is called protein sorting. There are two different types of protein sorting depending on if the proteins are being produced from free ribosomes in the cytoplasm or from ribosomes attached to the rough endoplasmic reticulum. Proteins that are produced on free ribosomes will be sorted to one of 3 or 4 locations based on the presence or absence of a specific signal sequence of amino acids called a signal peptide. If the protein does not have a signal peptide, it ends up in the cytoplasm. An amino terminal sorting peptide will send the protein to the mitochondria or the chloroplast of the cell, while an internal signal peptide will send it to the nucleus. Proteins produced by ribosomes on the rough endoplasmic reticulum end up within the lumen of the endomembrane system, embedded in the membranes of the cell, or secreted into the extracellular fluid. Proteins destined to enter the endomembrane system contain an amino terminal sequence recognized by a protein called the signal recognition protein, or SRP. This causes the ribosome to pause in its translation and bind to the rough endoplasmic reticulum. The ribosome then continues translation and the protein enters the endoplasmic reticulum by completely passing through a channel protein in the rough ER. These proteins become proteins that either reside in the endomembrane system or are secreted from the cell. Alternatively, the protein may pause as traveling through the channel due to the presence of a signal anchor sequence and becomes embedded in the endoplasmic reticulum membrane. These proteins will travel in the membrane until they reach the cell membrane, where they will function as receptors, pumps, and channel proteins.